Hello,
Hey, what's up?
1 second. Do you hear me?
Yep.
Sorry. So you got my questions I sent, right?
Yep.
Okay. Uh, let's get right into it then. Let me share my screen.
Did you take a new practice test since the last time we met? No, right?
No, so my next one I'm probably planning on taking
Uh-huh.
on Monday.
Got it.
That's gonna be like 9 days since my previous.
Mm-hmm.
Yeah. All right. Are you able to see it?
Mm-hmm.
Okay.
Yeah. So, uh, why don't you, yeah, walk me through how you would approach this?
Okay. Um,
So it says a monoatomic ideal
right.
gas is initially sealed within a container featuring a piston fixed in place at one end. The piston is then unlocked and allowed to move under constant pressure. How does the initial heat capacity of the gas at constant volume CV compare with the heat capacity of the gas at constant pressure CP, and why?
Yeah, I mean, I know ideal gases, there's like five main rules.
Okay.
I'm not sure that applies here, but like, I mean, yeah, I'm sure it does, but
Tell me about
um,
the rules.
what?
Oh yeah, tell me about those rules.
One of them is elastic collisions.
Okay.
Um,
Tell me about that.
it means that energy is not transferred
Mm-hmm.
from one collision to another.
Okay.
Um, the volume of the particles is negligible compared to the container.
Mm-hmm.
That's two. Three is there's no kinetic energy, or there might be. Like that, I forgot.
Yeah.
Or kinetic energy is the average of like all the particles.
Yeah. So that's gonna be um I guess like how to describe temperature, which is always going to be a thing. But for
Mm,
ideal gases, yeah, you're not going to be considering the volume of the gas particles themselves. Um,
mhm.
and then another like thing is that there's no intermolecular forces between the particles, like no attraction or anything like that. So, like, what conditions would enable an ideal gas to act, or would enable a real gas to act as an ideal gas?
High temperature, low pressure.
Exactly. 'Cause if it's high temperature, they're moving around so fast that they don't have a chance to interact with one another. And if it's low pressure, similar type of thing, they're kind of going to be spread out. But okay, so we have this piston thing happening, and then it's allowed to move under constant pressure, and then the heat capacity at constant volume is going to be something, and then the heat capacity at constant pressure will also be something.
I mean, when it says constant pressure, like we don't know if it's low or high pressure.
Uh-huh.
So
So let me see. This seems like okay, so let's see. So heat capacity at constant volume. So this is so something that happens in this type of situation is PV work. So like if I were to plot pressure and volume here, something like that. Um, the P delta V work involves energy transfer via delta V at constant P. So that's going to be, let's see. Okay. Initial heat capacity. Okay, so constant pressure. Then how does the initial heat capacity of the gas at constant volume, CV, compare with the heat capacity of the gas at constant pressure, CP? And then the choices say CP will be greater than CV because the gas can expend energy by doing work on the piston. CP will be less than CV because the movement of the piston will prevent heat transfer. CP will equal CV because the internal energy of the gas can no longer change. And then CP will equal CV because the gas molecules are monoatomic and ideal. Yeah, so tell me what you did here. Or yeah, what you're thinking as you do this.
um I mean it was a good go, but I wasn't fully sure like I know I mean we have the equation PV equals nRT like but that's it's like the furthest I could really get. Like yeah, like I think I put D, which was like wrong, obviously. Um so I don't know.
Okay, let me see. Capacity of the gas, constant volume of CV. Here the heat capacity, constant pressure. heat capacity of the gas at constant volume, heat capacity of the gas at constant pressure. And then which heat capacity will be larger?
I mean, I'm sure your capacity is like, it's gonna be like Q equals MCAT equation.
Mm-hmm. Yeah, like it's similar to that in that, well, specific heat is going to be where with that Q equals MCAT
Mm-hmm.
formula. Heat capacity is essentially the same thing, except you're not considering the mass. So
Mm-hmm.
it would just be Q equals C delta T. But I'm trying to think of like what the relationship must be or might be for the heat capacity when it comes to, okay, if I do this, I get something like that, which is heat over temperature, and maybe this could come into play.
I think it does.
Okay. Okay, so let's think about, okay, so I'm not sure if I should be thinking of this as like a heat engine or anything like that, but let me see. But if you have, okay, so if it's constant volume, then the gas wouldn't be doing any work because that volume doesn't change, hence no PV work because the delta V would be 0. Um, and so at, you know, constant volume, you will have, well, the gas won't do any work. And so all of the heat that's added contributes to like the internal energy of the gas, aka like its temperature. But I'll just write internal energy. How should I put that? I'll put like U with a little I subscript. So that would be for constant volume, heat capacity at constant volume. Then the heat capacity at constant pressure. Now, that could be something that will enable the gas to expand and do work against or to do work on the piston. So the heat that's added, so okay, at constant pressure, the heat that's added will be going into work as well as the internal energy. Internal energy is just like, like the I guess part of it would be like the temperature, I suppose. But okay, let's see. So they're saying constant pressure then constant volume compared to constant pressure. So I think that this then would have to be larger because it's not just contributing to the internal energy, but it's also doing work. So, and when you're doing work, all right, so let me, I guess, go over each of these. So A says CP will be greater than CV because the gas can expend energy by doing work on the piston. So for a lot of these, I like to just evaluate the second part first. And the second part of A seems to be something that happens. Let me look at B. Less than CV because the movement of the piston will prevent heat transfer. I mean, I don't think the heat transfer part is what's happening here. I don't think any heat's being transferred. CP will equal CV because the internal energy of the gas can no longer change. Well, it could change. CP will equal CV because the gas molecules are monoatomic and ideal. That doesn't really explain why CP would be equal to CV. So I would think of it perhaps like this with if it was, well, I guess like the formula that I'm thinking of is just what is it? The first law of thermodynamics, which is the change in internal energy is equal to the Q, which is the heat absorbed by the system, in parentheses, I can express that as something like enthalpy plus W for work, which is going to be PV work. So this is the first law of thermodynamics that that's the conservation of internal energy. So a change in internal energy of a thermodynamic system is going to be given by the sum of the work and the heat exchanged with its surroundings. So I think that would be the thermodynamic law to use here. And so yeah, and so I think that when it's at a constant pressure, since since the heat that you're adding is contributing to the internal energy as well as work, that CP must be larger than CV. So I think that would be it. Is that what the answer is?
I think so, yeah. At constant pressure,
Yep.
that means there's still a change in volume.
Oh, yeah, just because there's um uh yeah, yeah, essentially because at a constant pressure, well, I guess first of all I should say at a constant volume, like this volume does not change, so there can't be any movement of the piston, right? But if there's constant pressure, we don't have to have the same volume. We can have the volume change, which would cause PV work to happen. So the PV work thing is pretty much what, you know, this is mostly about. Like in a piston like of a combustion engine, right? You have like let's say like A, B, C, D. Like A to B involves um A to B involves a change in volume with roughly the same similar pressure, but then B to C is the pressure change at constant V. And then C to D, we have a reduction in volume at something close to the constant pressure. And then D to A, it's going to be a change in pressure. Pressure will go up at the constant volume. So the thing about this is that this part that's vertical and this part that's vertical, both of these have a delta V of 0, aka, you know, um, no PV work. And so that's happening when volume is equal, right?
Mm-hmm.
So in the condition where we have a constant volume, all of this heat energy is being used to heat up the internal energy of like the molecules, which means, the molecules will move faster and have a higher temperature, which means a higher kinetic energy, average kinetic energy. So when you're having a constant V, you're not having no PV work. So all of that energy contributes uh, only to the internal energy. Whereas I'll use, let's see, pink. So here and here, you are having a change in volume. So for the pink ones, PV work, is not 0. So work is being done there. So essentially, to answer this question, I would think of it as, well, the constant volume means that whatever heat you're adding, uh, it does not contribute to PV work. And so it's just going to go to the internal energy. But at constant pressure, which means that volume can change, then that means that you're going to have the heat energy being kind of split or contributing to both the internal energy as well as the work that's done by that piston.
Okay. The thing is, it says it's an ideal gas. Isn't like volume negligible?
Well, when we talk about ideal gases, as you said before, right, we're not using volume to equal the volume of the individual particles. We're using volume to indicate the volume of the container, essentially,
Okay.
right?
Hmm.
So so I mean, they drew a pretty good picture here, but I can draw it kind of again to be like, oops, to kind of show that, like, let's say this is, where the gas is, and then whatever, this is some type of piston system. So like if the piston, goes down, it compresses the gas. And if the piston goes up, it expands the gas. But we're still just talking about the volume of the gas container itself.
The volume is gonna But like if the piston goes up, does the volume increase?
Yep. Yeah. Well, I should say this. I let me kind of change what I just said right now. So if you have a constant volume, that is the case for when this is at constant volume. We can't say that the volume changes because the condition that they're giving us is constant volume. But what can change, right? Oh, and I should also say if you have a constant volume, you don't have any PV work. So essentially, maybe this is a bad picture, but let's say this is a gas. This is like, part of a piston. So if we have a constant volume, aka the volume of this container with a gas, if that does not change, right, then that means that there can't not be, because the type of work that we're looking at for thermodynamic systems like this it's called PV work, which is P delta V. And so if the delta V is 0, like it is in the case when you have a constant volume, right? We have a constant volume, that means that delta V is 0, and PV work will then be 0, which means no work is being done.
Okay.
And but if you have constant pressure, right, you could have the volume change. And if the volume goes up, it's going to press against that piston, pushing that piston outwards more.
What is pressure here?
In which case?
Like when they say like pressure, what do they mean exactly? I mean, volume is like the
Uh-huh.
volume of the container.
Yeah. Volume is the volume of the container, not of the actual stuff in the container, right?
Yeah.
Pressure is essentially, if you have, let's say, a high pressure, it will push against the piston, causing, so like if this goes, like the pressure, sorry, if the volume, sorry, if the pressure is constant but this volume increases, it will press against the piston, causing the piston to move up.
When it's constant pressure though, is it not moving up?
Oh yeah, so if it's constant pressure, that means that we could have a change in volume. And if there's a change in volume, then we have PV work. And if there's PV work, the system, which is, the little container here, will be doing work on the environment, which is the piston, pushing that piston outwards. It's kind of like a steam engine. Oops. Like a steam engine, right? Like you have let's imagine the gas molecules um are or let's imagine we just have in this container here with a piston water, right? And like liquid water. And let's say that we boil it, and if we boil it, the liquid water turns into, water vapor or steam, and the steam is going to have a larger volume than the liquid water, and that larger volume will cause PV work to happen, and it will push this piston upwards. So if you imagine just doing that with, like, take some water, put some, like, I don't know, if you like boil water or whatever, and you put like a whatever, like a
I know exactly what you're talking about. Yeah, when it boils, it
oh, and like a
like
pressure cooker.
expands.
Yeah, and a pressure cooker works that way too. So, oh yeah, good. It is a, yeah. But Yeah, okay, let's see what they're saying here. Okay, constant pressure, you could have work done on the gas. You could also have work done by the gas, right? Like for instance, in this case where, or I'm gonna erase what I wrote. So like in this case here, we have, what is this? Ice melting or something. But yeah, whenever you have constant like basically what you're looking out for is if there's constant volume or not. If there's constant volume, delta V is 0 and PV, aka P delta V, work will be 0. So constant volume, no work is being done. Constant pressure, though, work can be done. And it could be done in two different ways. It can be work done on the gas, which would cause the piston to go down, or it can be done by the gas, which will cause the piston to go up. And so I guess what they mean here by the ice cube is that when it's cold, right, the particles will be moving less quickly and won't be traveling as far. So that will cause that, piston to go down. But if they're heating it up, like in the example I gave for the steam engine, the molecules will be moving faster and they'll be hitting against the walls of the container, causing an increase in well, I was going to say causing an increase in pressure, but they're saying constant pressure here, but it's only going to be constant pressure. I guess that's also important because if we had this system here, whoops, with these the gases here, if these gases expand, right, it will cause a higher amount of pressure in here. And to relieve that pressure, the piston will have to move upwards. Right?
Mhm.
So yeah. So basically, that's what you want to look for. So basically, if you have, like, I would look at it like this. If there's constant volume, there's no PV work, piston isn't going to move, right? But if you have constant pressure, really, I think of that as changing volume. And if there's changing volume, there will be work done, PV work. And then it's just a matter of, is the piston going to go down or, up? So if the piston goes down, the work, or rather the surroundings, everything in thermodynamics is like surroundings versus system, right? So if the system is the gas, the surroundings will be doing work on the gas if it's, you know, if the volume goes down like this. But if the volume goes up like this, then the work is being done by the gas, like on that surroundings. But yeah, let me see what else I wrote here.
How do I know when to like use Q minus W or Q plus W? I always like mix it up.
Oh, okay, that's a good question. So, um, I like to think of it as, I mean, I generally always I prefer adding rather than subtraction. Um, but first of all, you need you do need to be aware of what happens. Let's see. I got like a nice picture for you, but yeah, it's always something that has to do with here. Okay, let me see. Because yeah, there's like, so basically when you are, let's see what I can just write here. Okay, so if the surroundings are doing work on the system, it'll be a positive work. And if the system is doing work on the surroundings, generally you're going to have just the perspective of the system's perspective. So yeah, if the system is what's doing work on the surroundings, it'll be negative W work. And if the surroundings are if the system is absorbing heat from the surroundings, it'll be positive Q. But if the system is producing heat that gets absorbed by the surroundings, it'll be negative Q. So in the yeah, in that equation, I prefer to write it as Q plus W, but of course that would change depending on, you know, if it's the system that's doing the work or absorbing the heat or the surroundings, right? But I would prefer to do it like delta E for the change in energy is Q plus W. And let me see what they wrote. They wrote something like, oh yeah, here, Q minus W. So here we have a positive Q, which means that the surroundings are doing or adding heat. Let me see. Q minus W. So okay, so if I have a positive Q here, I'm gonna have this as heat absorbed by system, and then this positive W will be work done on the system. So in what they wrote here, they do have heat absorbed by the system, but the negative W indicates that the system is doing work on the surroundings. So you know what? It's actually just this formula, and everything else depends on your situation. Now we know from the situation here in the question here that they're telling us that the gas is doing well at constant pressure. The gas is doing work on that piston, meaning, and the gas is our, you know, system, and the piston is like our surroundings. So if we have that work done by the gas like this, that's going to give us a positive. Oh crap, I mean negative.
No, yeah, negative. That's expansion, right?
Yeah, yeah.
Expansion, the gas is expanding, like the piston.
Mm-hmm.
Like it's going up, so it's doing work.
Yep.
And then when the system, basically the surroundings are doing work on the system, it's compression.
Mm-hmm. Yeah. So generally, if something's being done on the system, you'll have it could be positive work, it could be positive Q. or energy transfer, heat energy transfer. But if it's being done by the system, we use negative values.
All right.
Yeah.
What's the next question?
Okay.
So talks about compressibility. Um, Real gases exhibit non-ideal behavior because molecules occupy a finite molecular volume and intermolecular attractions present between molecules draw them closer together. Compared to the volume of the ideal gas at 358 atm, the volume of an equimolar amount of real gas under identical conditions is.
Okay. Oh yeah.
Okay.
Okay. So how would you go about this?
Uh, I mean, we know that. Let's see. So compressibility is PV over nRT.
Okay.
So, well, at 350, well, it's like 1.2.
So if this PV over nRT is equal to 1, that's an ideal gas, right?
Yeah, so that's at 250 atm. Um, so, well, as pressure increases, volume decreases,
Sure.
right?
Yeah, yeah, they are inversely proportional. Um, but let me see. So compared to the volume compared to the volume, wait, so 200, okay, okay, compared to the volume of the ideal gas at 350, which is 1, and then here it seems like it's like 1.15 or something. The volume of compared to the volume of the ideal gas at 350, the volume of an equimolar amount of the real gas under identical conditions is so um okay, this is like the question a question that asks about like the deviations. So if PV equals nRT is larger than 1, it's well going to be something like um a higher volume, because okay, so let's see, we know that the volume of the volume of a real gas compared to the volume of an ideal gas, which is larger?
Okay, is it a real gas because like you talk about the volume of the container and the particles?
Uh-huh. Yep. So you should have a larger volume for a real gas compared to the volume of an ideal gas, right?
Yeah.
Okay. And then if it's less than 1, so we know the volume kind of difference for the for the real gas. What else would change? Maybe something like pressure.
So why is it pressure?
So we know that if it's high pressure, so you know how a ideal gas can behave as a real gas if it's at high temperature and low pressure.
Yeah.
The high temperature part is just so that there's no chance for the molecules, to have intermolecular forces. But the low pressure also assists in that because you're not really having the individual molecules of the gas interacting with one another. So that could be expressed as low pressure,
Hmm.
right? So um yeah. So basically, also the volume, the molecular size of, like, that volume assumption is also dependent on the molecular size being significantly smaller than the intermolecular um separation. Like if this was a molecule and this is another molecule, the distance between them is going to be much, much smaller, or rather, the volume of the two gas particles must be significantly smaller than the spaces between each of the gas molecules.
Okay.
But but yeah, like essentially, like what helps me remember is just I'm thinking of like a gas in space. And if it's a gas in space, you could have the gas expand tremendously. And the gases that expand tremendously in space won't have the particles interacting with one another. So, so that is reflected then in the pressure, where the pressure of the real gas is smaller than the pressure of the ideal gas. And it kind of also makes sense when you consider how P and V are inversely proportional, right?
Hmm.
And so, okay, so now, so they're saying that compared to the volume of the ideal gas at 350 atm, the volume of an equimolar amount of the real gas under identical conditions is, all right, so we know that, are there any of these choices we can eliminate?
A.
Yep. Can't be the same. And what about B?
No.
Tell me why.
Um, just because I think it's C. I can't tell you why it's not B though.
Okay, okay. Try though. Try it. Try it.
Yeah. Um,
And I think it's best to read the stuff after first.
mhm. Well, moles,
Yep.
number of moles is M.
Yep.
Um, I don't know.
So it says
Um,
because the number of moles of each gas is the same, but their molar masses are different.
if the number of moles is the same, does that mean mole mass are the same?
No, but what is there anything here that's telling us that there's a difference in molar masses between like the ideal condition and the real condition?
Yeah, there isn't.
Correct. So nothing is telling us that their molar masses are different. right?
Mhm.
Okay. Now, what about since you said C, let's look at D first. So D,
Um, well, the intermolecular interactions that has to do with temperature. I I don't know. Like, doesn't have to do with
mhm,
what we're talking about.
uh-huh. Yeah. Uh so let's think. Okay, is the intermolecular interactions between the gas molecules significant or not? Well, if they are significant, that should give us. So I should also say that this over here, the pressure, the pressure of the real gas is smaller than the pressure of the ideal gas. This is due to the intermolecular forces assumption, right? So if that was the case, that the intermolecular interactions between the gas molecules are significant, we should have a PV over nRT value that's smaller than 1, right?
Honestly, I'm still really confused because,
Mhm.
like, don't real gases function at high pressure, low temperature? So I don't understand, if an ideal gas is high temperature, low pressure, how does the pressure of an ideal gas end up being higher than the pressure of a real gas?
Okay. So, uh, okay, so let's think about the overall kinetic whatever theory of ideal gases. So, a gas particle is just this hard spinning, moving, uh, randomly and constantly colliding with each other and with the walls of the container. I think of like pool balls, you know.
Yeah,
There's no like compression or something when they hit each other, they maintain their shapes. Um, there's no, yeah, deformation or anything like that. And so the assumptions would have to be that the molecular size must be significantly smaller than the size or the length of the intramolecular separation. That's one. Two, intermolecular forces are negligible. Three, all collisions are perfectly elastic, which means both momentum and energy are conserved because between colliding objects. Um, Energy exchange between particles is solely due to the elastic collisions, and that the molecular speed distribution is constant in time. Um, and and a caveat to all of this is that the identity of the gas is not important. Um, Even though gases can be quite different, that's not going to play a role in whatever we do with ideal gases. So that's kind of what would be a reason to eliminate B, because if they're saying the molar masses are different, they're implying that you're using, you're using a gas that, or you're using two gases or something with different molar masses, but the identity should not change. Like you could have gases of varying molar masses, um, but that doesn't play a role. So what does that mean? It means that real gases behave like ideal gases at very low pressure and very high temperature, because that will make the relative volumes and intermolecular forces negligible. Um, and that means also that the greatest deviation from ideal gases in terms of, oops, in terms of behaving like real gases would be when the temperature is really low and the pressure is really high. The pressure being really high means that the particles are more likely to collide, and the temperature being low means that they're going so slowly that there could be intermolecular forces at play. So for this question, they're giving us a temperature of 200K, whatever, and they're giving us explanations for how real, or for the circumstances for a real gas to exhibit non-ideal behavior. And that is if the molecules occupy a finite molecular volume and intermolecular interactions draw them closer. And then they're saying compared to the volume of an ideal gas at 350 ATM, the volume of an equimolar amount of the real gas under identical conditions is, so we eliminated same because it can't be the same. We eliminated B because if the molar masses are different, it's not going to change anything. And then C and D, and then for D, it's says larger because the intermolecular interactions between the gas molecules are significant. Now that is this case here, right? If the P of the real gas is smaller than the P of the ideal gas, which is the case because we see that this is the condition to behave as a real gas, right?
Mm-hmm.
Um, so so yeah, if the intermolecular, so like the second part is true, right? Because an ideal gas, sorry, a real gas will have those intermolecular interactions. However, if that was the case, it should not produce a larger volume, right? It should produce a smaller one. So this shouldn't be our answer here. And just because the intermolecular force assumption that the second part is talking about should result in the PV over nRT as being less than 1, right? And that's because, well, so like if you do like P real volume over nRT, right? It's going to be less than P ideal V over nRT. And so so okay, does it make sense that a real gas will have a smaller pressure?
I guess, like kind of. But if it doesn't, why is it still greater?
Why is it still greater?
Yeah.
You're saying for the,
Like you're saying the ideal gas will have a small pressure, but it's still greater than the real gas.
oh yeah, the ideal gas will have a larger um pressure, which is why like um wait. So wait, sorry. So you're asking why does the ideal gas have a higher pressure?
Yeah, because you said the ideal gas has a small pressure, but like it's a small pressure, but it's still greater.
Oh, I see what you're saying. So let me kind of take a step back then. So gases, so the two main things that we're looking at are the intermolecular interactions uh and the volume. And so for a gas to behave as a real gas, you should have a like a low temperature and a high pressure. And oh, I see what you're saying. You're saying why is the ideal gas pressure larger if the ideal gas, uh, or rather, if the ideal gas or a gas behaves as an ideal gas when the pressure is very, very low, right?
Yeah.
I see, I see. So let's see. Um, so if we know that let's see.
I'll be honest, I just don't want to spend the whole session on this.
Sure, 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 sure,
Like
sure.
maybe I can rewatch what you said already later. I just, you know what
Yeah,
I
yeah,
mean?
yeah.
It's just like,
I'll just go right into this one for C. Um, Larger because the combined volume of gas molecules is significant relative to the volume of the container. And so that's what we have here, right? With the volume of the real gas being larger than the volume of the ideal gas. And it does produce a larger volume here.
I mean, I get like C, I get it was just more about pressure, but like I just don't want to keep going on almost in circles. Like I'd rather just rewatch what you already said.
Sure, sure, sure. Yep.
So just to get more done. All
Yep.
right. The other thing, I didn't send you this. I'd rather do this passage and like if we have time, we'll go over the like I sent you a third question on Doppler effect, but like whatever.
Sure.
Um, so this is it's like a genetics passage.
Yep.
Okay. So I guess I'll read it.
Okay. Yeah, read it and tell me whatever buzzwords and stuff you see.
Understood.
Yep.
The fruit fly DM is frequently used as a model system to investigate neurological disorders. Maybe neurological. I don't know if there's any buzzwords, but
Yeah, not really.
yeah. As in humans, male flies have a single X chromosome and female flies have two X chromosomes.
Yeah.
Um, single X, two X, like males, single X.
Yep.
Just saying that,
Mhm.
similar to humans. Three genes, ABL, enabled DNA, and receptor protein tyrosine phosphatase, PTP, are involved in axonal guidance during neuronal development and are all believed to operate in the same pathway.
Okay.
So just highlight the three genes
What's a
and we'll
phosphatase?
Hmm?
What's a phosphatase?
It adds a phosphate group. Just like a kinase removes it.
Well, a kinase is a transferase, so a kinase can add or remove, but a phosphatase is, well, a kinase just transfers, a phosphorylase adds, right?
Oh, okay.
And
Okay.
and adds through dehydration synthesis, and then a phosphatase will remove uh, through hydrolysis.
Okay.
But that's a buzzword right there. But get like the enzyme classes, make sure you have those down.
Okay. Um,
Yep. I'm putting a image in the chat for you to look over. But yeah.
this is like a classic image. I feel like
Yeah,
you
no,
should I like watched one or two sessions and
yeah.
I sent it to me already. I saved it, yeah. Thank you.
Yeah, yeah. For some reason, they
Um,
don't even list kinase for transferase, but whatever.
mhm.
But yeah, yeah. Okay, cool.
Okay. Um, okay. To investigate interactions between PTP and either ENA or ABL, fly lines with dominant mutant versions of each gene were developed and identified by eye characteristics. Um, so dominant,
Mm-hmm.
maybe.
Okay.
A dominant mutation that inactivates PTP results in red and white eyes. A dominant ENA mutation yields orange eyes. Yeah, so there's just all this stuff. It was like
Yeah.
information overload.
Yeah, this is when the proportionalities kick in.
Yeah. So, okay. So dominant mutation.
Mm-hmm. Yeah.
I mean, so how would you write it? Would you write 1 over PTP, the symbol red and white eyes, or like,
Um, I think maybe I'll read a little bit more because I see that the P in the parentheses is like the type, like whatever, like the type of the mutation perhaps, but yeah, I don't know if I would write anything yet, but you do have the right idea of like inverse proportion if you're inactivating it causes red and white eyes, etc.
okay. Um, Using these phi lines, the following experiments were conducted. The recessive wild type forms of each gene were indicated by lowercase letters. Okay. Female homozygous for wild type ENA and heterozygous for PTP were crossed with male heterozygous for ENA and homozygous for wild type PTP. Males and females from the resulting F1 generation with the genotype EEPP were then selected and crossed with each other to produce F2. And so then it tells us 2 mutant ENA alleles, as well as 2 mutant PTP, and at least 1 mutant ENA did not survive past the larval stage.
Okay, I'm gonna just see if I could write anything. So let me see. The a dominant mutation that inactivates PTP, parentheses P, results in. So maybe I'll say P inversely proportional to PTP and proportional to red, white, and E dominant. Okay, ENA doesn't really say inactivates ENA, so I'm gonna stand by on that, and I'm just gonna say um orange. And then maybe I'll say P plus E yields um, either red or orange. And then we have ABL, so A, which is, it's again, it doesn't tell me if it's inactivating it or not. Um, but we get elongated eyes. And then they did, okay, then lowercase letters for the recessive stuff. And then experiment 1 females homozygous for wild type ENA and heterozygous for PTP. So little e, and big P, little p were crossed, okay, with males heterozygous for ENA and homozygous for wild type PTP. Let's see. So I mean, that's going to be kind of annoying because it's a dihybrid cross, but you know, hopefully they won't ask us to. Sorry. Um, too much about that, but let's see. They're just talking about. So okay, I'm gonna write like EEPP times capital E, little e, PP. That's the parent.
Hmm.
And then some type of F1, males and females were then selected and crossed with each other. So with the genotype E little p times this, I believe, to produce an F2 generation, and then flies with two mutant ENA alleles, capital E, as well as flies with two mutant PTP alleles and at least one mutant ENA allele. Did not pass. Okay. So the ones that, so we'd probably have to do some type of cross for these, and whichever ones in the F2 generation have, like, I don't know how would write this, but if from the F2 generation we have anything with two capital E's and anything with like either two capital P's or capital E's and capital P's, they die. So we don't have to worry about those ones. But so far, what I'm thinking of is it's kind of like I'm thinking like, okay, dihybrid crosses could be pretty tough, and I'm just thinking I hope I hope that the questions don't be too hard. But yeah, I would just write that, I guess.
Okay.
Yep.
Um, so we read experiment 2, right? PTP and AB are located on the same chromosome. Females with a PTP mutation on one copy of the chromosome and the ABL mutation on the other were crossed with wild-type males.
So what does that indicate to you where when it says located on the same chromosome?
Length.
Yeah. I mean, I don't know if they're close enough to be
Yeah,
linked.
yeah, yeah.
That is
Okay.
what but good, because that is what you should be thinking. Yeah. Yep.
Um, okay, it tells us something being crossed. Um, in Drosophila, the recombination occurs only in female gametes.
What what's the significance of what's recombination?
Mm, like crossing over
Mm-hmm.
at prophase.
At prophase?
Yeah. Or is it metaphase? Sorry.
Yeah.
Metaphase.
Mm-hmm.
When they like line up. F1 progeny with parental and recombinant genotypes are shown in table 1. So, but if it says it occurs only in female gametes, like what's so relevant about that? That it's honestly, I don't know.
So tell me a little bit more about recombination. Like what is it? You said like metaphase, but in what? Is it mitosis or meiosis?
Meiosis.
Yep. And in meiosis, well, actually I'm thinking of like perhaps well, I'm thinking of like actually like after fertilization, but not sure yet, but that's what I'm thinking of. Just because prior to that they say cross with wild type males. Um, and I'm guessing like because only the nucleus from the sperm comes in. That's just what I'm thinking of. I don't know yet.
Okay.
Mhm.
F1 males with red and white elongated eyes. Were then crossed with homozygous BTP mutant females expressing normal eye shape. This produced an F2 generation in which 50% of both male and female progeny had elongated eyes.
All right, and then we see these phenotypes of 500 F1 flies. Now, let's see. So this could be kind of useful. So like white, normal sized eyes. That's like something with P, white elongated eyes. something with P and A, red and white, which is also P, normal sized, okay, red and white elongated, P. White and red seems to be the same thing, right? But okay, whatever. And A.
PA. Yeah.
All right. And then they just took the F1 males with red and white elongated eyes, and crossed them with homozygous BTP mutant females expressing normal eye shape. And this produced an F2 generation in which 50% of both male and female progeny had elongated eyes, aka the mutation in the A or whatever. So, okay, cool. All right, let's see. So experiment 1, approximately what fraction of the F2 generation is expected to die during the larval stage? Okay. So I guess it's good that 1/2 isn't a choice. Because I feel like if you just read this, actually that wouldn't make sense anyway. Okay, so yeah, we have to find the ones that didn't survive, right?
So this is, yeah, experiment 1. We would have to cross E.
Yep.
So we'd have to cross F1. So E, those two same ones.
Mhm.
Okay.
Yeah, so if flies have 2 capital E's or 2 capital P's, they will not survive. So I'm going to start by doing like a cross for the F1 to figure out, what percent will give me like capital E's and what percent will give me capital P's. So if I had E, lowercase e, lowercase e, I have capital E, capital E, capital E, little e, capital E, little e, little e, little e. So here it seems like 1/4 of that. And then if I do the capital P, little p, capital P, little p, kind of exactly the same. P, little p, big P, little p, little p, little p. So that wait. If the 2 capital E1s, okay, so it could be the 2 capital E1s here, so 1/4 times, and then it could be anything, capital E, as well as flies with 2 mutant PTP alleles and at least one mutant ENA allele. So at least one. Okay. Is this how you were going about this?
More or less. I also forgot how to do Punnett squares fully, so like that
Like
didn't help.
regular Punnett squares or like crazy diagonal?
Or I mean those. I mean, I think normal ones I can do them, but like yeah, like when it's two different things, two different genome.
Yeah, it could be that could be very annoying. So I like kind of split them up like this, but let's see. Okay, so this should give me 1/4, 1/4. Okay, if we have all right, I'm just going to do okay, I'm just going to do specifically what I have, or maybe F1. Okay, so parent generation is this, and then it can produce F1 that seems to be big E, big P, little p. And then for the F2 generation from this cross, we can have combination of big E, big E. All right, forget this for now. Big E, big E. And then from here, big P, big P, big P. We can also have big E, um, big P, little p. We can also have big E, little p, little p. We can have big E, little e, big E, little e, big P, big P. We can have big E, little e, big P, little p, big E, little e. Sorry, big E, little e. Um, big P, or rather, little p, little e, big P, little e, big P, little p, little e, little p. So, okay, so this should be all of like it's a lot, but this should be all. I think this should be all of the 1, 2, 3, 4, 5, 6, 7, 8, 9.
Yeah.
Okay, and then I'll use a different color for the like so the ones that die are anything that has capital E. So that like two capital E's. And then anything with two capital P's will also die. So that okay. And then okay. So we have,
This
yep.
do you take into consideration that like there's two big E little e and two big P little p? You know what I mean? Like it's 50%, but that doesn't matter.
Sorry, you're saying, am I taking into consideration that there's two big E, and there's two big P, big P?
Like there's a 50% chance
Huh?
of big E little e. There's a 25% chance of big E, and then 25% chance of little e little e. Honestly, whatever. Like just forget what I said. But okay, let's say we cross them out. It's gonna be 4 out of 9. Oh wait, 5 out of 9? No, 4 out of 9.
It said 4 out of 9. 1, 2, 3, 4. Okay, I'm gonna just do the ones that are big P, I mean big E, big E, big P, big P. Because there's supposed to be 16 of these in a 4x4. So big E, big P, or like this one, um, would be 1/16. Um, big E, and then this, man, I hate how it's like I can't tell almost like my big P,
Yeah, I got you. It happens to me too.
but I think it's these two. So big E, big P, little p should be 2/16. And then big E, little e, big P will also be, so big E, little e, big P, 2/16. And then big E, little p, this guy, little p is going to be 1/16. And then I have, wait, is there anything with the big E, big P, little p? Oh, yeah, I think it's that one. And that's 1/16. Um, okay. Yeah, I think these should be it. Um, and what do we do with these probabilities? Do we add them? Do we multiply them?
How'd you get the probabilities?
Oh yeah, so I just wrote out like over here, I wrote out
Yeah.
the different genotypes. And then I just counted like if there was one instance of it, then I just made it 1/16 possible ones. And because there's supposed to be 16 total, right? And the ones that I wrote are the combinations for the F2 generation here. So so yeah, essentially I just wrote out all the possible combinations. So like big E, big P, then big E, big P, little p, and then big E, little p. So that's that. And then big E, little e, and then I do the thing with the p's being big P, and then big E, big P, little p, and then big E, little p.
I get that, but why is like
Yep,
capital E, and then big E, lowercase, whatever. Why is that 2/16?
the 2/16 for these two.
Yeah, like what
Uh-huh.
I think I get the bottom one. Why is big E, big P, lowercase, little p,
Hmm.
2/16?
Yeah. So because there's two of those genotypes, right? Because you have in your F, so like your parent generation here, right, produces the F1 generation here. And you see how there's like big P that we're crossing.
Yeah.
And so when you cross that, you'll end up with like big E, like um big P. You can have big P, big E, sorry, little p, right? Kind of like here and there. Like this is the like F1 cross here between the two heterozygous ones. which means
Yeah.
two of them, right?
Okay.
Yeah. So yeah, now we just add up these um yeah, these probabilities. Oh crap, I just realized what I just said over here, or rather what I said over here, there's two of those with this. So basically we have all of the basically we have like um big E, and then the three different types of P's. Because anything with two big E's is going to be not passing it to the larval stage or passing the larval stage. Um, so I have that. So you can see, for instance, this, and this all have two big E's, right?
Yeah.
That this one has two big P's. This one has a big P and a little p that we should have two of. And then for the one that is big E, and little p, we have one of those. So that'll be the one, um, the 1/16th one, right? Because it has the big E.
Mhm.
And yeah, and then for this one, it's big E, little e, and then big P. Sorry, it looks bad. Big P. So this is the one where it's not going to be the one with the two capital E's, but something like this, right? With two big P's, right? And yeah, any of these that has like a heterozygous thing, like here we have big E, little e, and here we have big P, little p, anything like that there's gonna be two of them.
Okay, so that yeah, that answer like that's what I was asking. 'Cause it's
Yep.
like 50%.
Mm-hmm. I see what you're saying. Yeah, when you're doing like the cross like this, yeah, you could see how these, right, are 50%. Yeah.
Okay, so we would add them all.
Yep. Yeah. Um, yeah, you could always do the dihybrid cross, the full thing, but I think this way is a bit faster. But yeah, you'll add them all up, and then you should get one, two, well, four, five, 7, 8 out of 16, which is what, one over, wait, what the hell? One
I'm
over two, right?
something.
Wait. Hold on.
So,
Oh, maybe it's
hello? So,
okay, no, I realized that um it really should be, I'm going to use red. It really should be this. It should be big E, big P. There's one of those. Big E, big P, little p. Two of those. Big E, little p. One of those. So this handles all of the ones with big E and big E. And then
Yeah.
it's big P, big P. So then that would be big E, little e, big P, big P. There should be two of those. So this is 6 over 16. And that's just going to be 2 over, oh no, 3, should be 3 over 8. So
Okay, wait,
the way that I would do this is write out all of the genotypes with 2 capital E's, and then the remaining genotypes with 2 capital P's. That's how you would do this.
mhm.
So is that it? Is it 3 over 8?
Let's see. Yep.
Yeah. Yeah. So yeah, the dihybrid cross like this it takes too long, but what I ended up doing was I from here it says anything with big E, right? So I'm gonna take big E, and then every possible combination of the P's. It could be 2 capital P's, it could be capital P, it could be little p, right? So that's 1, 2, 3 already, right? Oh, well, this one there's 2 of,
True.
but yeah, yeah,
Okay.
yeah, yeah. So that should, yeah, okay.
Okay. Um, next.
Mhm.
It asks for which one is autosomal.
All right. So what does that mean?
It's not sex linked.
Um, Kinda, sex linked. I think you're trying to say like it's not in a sex cell.
So it's somatic cell.
Um, yeah, so yeah, I think you mean like not in a gamete, right?
Mm-hmm.
Okay. Autosomal. So yeah, what are your thoughts here? I'm thinking maybe the thing about the same chromosome thing could play a role here, but
Mm-hmm.
well, yeah, what are your thoughts here? Actually, I don't even see much of a difference between these.
Um,
Because they say recombination.
okay, so I mean, yeah, so recombination happens in female gametes.
Uh-huh.
Combination happens in not non-autosomal genes.
These things are involved in axonal guidance during neuronal development, right?
Okay.
Is it all of them?
Yep.
Uh yeah, so I just looked at, here where they say that. Well, let's see what they say. Autosomes, same proportions. Okay. Oh no, you're right. The opposite of autosomal is sex linked. I thought it just meant that it happens in the gamete, but it's yeah, you had it right. Okay.
Wait, but hold
Yep.
on. So um it talks about experiment one
Mhm.
and ENA. Like how does that relate?
Uh, let's see. Okay, so I guess they're saying that since all of these genes that they're talking about um have two alleles, right? Then that would mean that it would have to happen in like a diploid cell, not a haploid gamete cell. Because if it was like, in a gamete, you would only each cell, like, sperm cell or egg cell would have to have only one copy of that gene, right? Or one allele.
Mhm.
So yeah, if it was just one allele for each of the cells, um, then yeah, that would be, on the oh, and then they say that up here in the top two, like males have a single X chromosome and female flies have two X chromosomes. Um, so we get to see that there isn't, you know, because like the idea behind sex linked down here is that since females have two X chromosomes and males have one X chromosome, whoops, that you should see sex linked traits that are recessive present at higher rates in males because they don't have as much of that gene. So here it says because males and females all have the same number of autosomes, autosomal traits are generally expressed in the same proportions in males as in females. So the fact that females have two X chromosomes and males have one show that there's that females have a larger amount of you know chromosomes in the sex chromosomes, uh but we're seeing here that that doesn't change the it doesn't change the inheritance pattern, so they should be autosomal because it's what you said essentially about it not being sex linked.
But but you thought it was the last one because of it was axonal guidance?
um oh yeah, I thought it would be because they mentioned how it's playing a role in axonal guidance, and I thought that it would be an autosomal cell, um, because of that, because it's not in the gamete. But I think that's not particularly the best type of thinking for this. I think you were right about thinking of this as whether it's sex linked or not, and that the entire idea behind sex linked is that if it's a recessive allele, that women would since they can have a second X chromosome, a recessive thing would only, like, unless they're homozygous recessive, uh they'll if they have just one copy of the recessive allele, then they can just be carriers of something. Whereas for males, they only have one X chromosomes, and that means that if it's a recessive, disease or whatever, that they don't have that second X chromosome to kind of cancel it out. So you should see that type of pattern of inheritance where males have, more of those sex-linked recessive traits. Um, and since they're telling us here that it's the same thing in fruit flies, males have a single X chromosome, females have two X chromosomes, that if it was sex linked, you should see this type of issue um, being overrepresented at for the males, right?
Okay, I got you.
Mhm.
Next is this.
Okay. This is kind of like what you're talking about when you read it.
Yeah,
All right. What do you think?
table one. So
So PTP, or if you inactivate, I'll draw this again. That, and then able. Elongated eyes. So tell me the consequences of being close together on the same chromosome.
there's a higher chance they cross over together.
Mm-hmm.
Yeah.
Okay.
So PTP, red and white, A is elongated. They cross over together, that means red, white, and elongated,
Yeah.
which is, yes, that's the recombinant. So
Okay.
then would it be B?
Let's see. Only a small percentage of the progeny are recombinant. Red, white, elongated, recombinant, only 21. Red, white, or rather, well, there's no other red, white, elongated. Um, yeah, I think that would make sense.
But
Yep.
yeah. Progeny. But let's just eliminate everything else. The
Yep.
two recombinant genotypes occur at similar frequency. So is what is the other? Oh wait, similar frequency. Wait, but so the top one it says recombinant white normal eyes.
Mm-hmm.
So like I don't get that. Why is that one recombinant?
You said red and white elongated eyes. Why is that recombinant, or the white normal?
No, the white normal eyes.
Okay. Um, yeah, I was trying to figure out like because they're mentioning something about red and white eyes, and then orange eyes, or red and orange, but I didn't see anything about just white. Um, so it's kind of weird. Do you see, like maybe I missed it? Did they say anything about white eyes having something to do with anything?
No, I mean I was looking for that too, yeah.
Yeah, I think it may not. I think so. So okay, so we know that they're on the. So if we know that they're on the same chromosome, we know that they should. Let's see. They should be recombinant or okay. So the two recombinant genotypes occur with similar frequency. Um, so the recombinant ones we see similar frequency, like 22 flies for the white normal sized eyes and 21 flies for the red and white elongated eyes. That's showing that there's a similar frequency for the recombinant genotypes. Um, but not sure if that would answer the question about um, like the distance between the genes. Um, and I think that what can really help us out is just how the recombinant ones have such small values for the number of flies. And then for like A could be something that's true, but it doesn't really answer the question, I think. Um, yeah, and then recombination did not occur in male. Yeah,
Wait, okay. Wait 5 minutes.
sure.
Um,
Yep.
yeah, I wanted to go over this one.
Okay.
So like here we basically have to work backward. I was just wondering how you would go about this question.
One sec. Okay. Um, suppose a female heterozygous for both PTP and ENA, okay, was unintentionally mated with a single F1 male of an unknown genotype from experiment 1. If 25% of the resulting F2 progeny, okay, died in the larval stage, that means they need to have like either big E with any combination of P's or big P with essentially any combination of E's. And that is 25%, okay, of the F2. So okay, let's see. Big E, little e, big P, big little p, and then I erased what I wrote for the F1 male genotype. Let me see. Wait, it's just that. Okay, unknown genotype of the F1. And 25% died in the larval stage. Genotype of the F1. Okay, so this times something should give us either something like that with any combination of these, or any combination of E's with big P's. Oops. Okay. Let's see. So anything with big P, sorry, anything with big E's, anything with big P's. Quarter of the progeny died. Non-viable combinations. Let's see. Okay. So all right, it can't be that. Wait, can't be that because that's not going to give you any uppercase E's or uppercase P's. C, so I'm eliminating C. If this was the case, let's see. %25 died. Okay. So okay, I'm gonna do capital E, little e for this female, and I'm going to test out these choices for the E's. So if we had big E, little e, it will be this. 25% over there. But I could also do the thing with, well, if I did little e, I wouldn't have any, oops, any big E, right?
Yeah.
So that should be out as well. Now, just evaluating the P's. So the mother has big P, little p. And now I'm going to compare little p, and big P, little p. I mean, I'm pretty sure it's going to be D, but let me see. Little p, big P, little p. Basically, nothing here would give me two big P's, right? If I cross big P with little p. Right. So A should be out. And then lastly, it should be D, but I'm just gonna make sure big P of this female, and then big P of this male gives me big P, big P, little p, big P, little p, little p. And then we have our 25% here.
Wait, no, isn't it capital P,
Gonna die?
twice?
Oh, would you say twice?
Big P is twice because, right?
Oh crap, what did I do? Okay.
Yeah, because yeah.
Okay. Big P. Okay. So that's this and this. So it would need to have both of that. Or so that could be the 1 over.
Yeah, I mean, I don't get how I'm supposed to do this under time constraint. I mean,
Yeah.
MCAT question, no way it's gonna be this hard.
Yeah.
No way.
Hybrid crosses are really annoying. That's why I guess I try to do it like part by part. But when I do it part by part, I also have to make sure that I put it into terms of over 16. So I think this would be something like, let's see, I don't know, something like that, somewhat close to the 25%. But so okay, on the actual thing, when you have a time crunch, let's do it like this. You know that you need to have either two big E's and doesn't matter what the P's are, or two big P's and it doesn't matter what the E's are. And if you have two big A's and two big P's, that could be one genotype. If you have two big E's and any combination of P's, that's another genotype. And then if you have two big P's and any type of E's, that's another genotype. So if you're able to at least recognize that those are the genotypes you need, then it can help you eliminate at least B and C, because B is not going to give you that. And C won't give you that either, because B has two lowercase e's, and you're not going to get a big E with that. C has lowercase e's, you're not going to get a big E with that. So you can eliminate B and C, and then when you're between A and D, where you have A as big E, little e. Well, A and D both have big E, little e, but D has big P, which would cause, the non-viable offspring. So on a time crunch, you will at least be able to eliminate B and C, and then with 50% between A and D, you can just I guess, use your judgment at that point. And also like the 25% of them that died, I think that's where it would take too much time. So I would just look at A and D and think about which one is most likely to have the type of genotype that would cause them to die, instead of thinking what is going to
Yeah,
25%. So, you know,
it's A.
it's hard. Okay, well, I suck. Let's see.
Honestly, I don't even know how. Like, I don't get that. Oh, because I mean, I understood that it would be D.
Mhm.
Oh my God.
It's okay. I can do. I'm going to take a screenshot and I'll just look at it after our session and get back to you with it. Because what I really didn't want, oh, they used like a pedigree chart as well. But yeah, because I was trying to look for what would give me like the most amount. So if it was the case that if you go down a bit, I want to see if they wrote for D, if it's more than it is or not. Okay, 25% would die. Okay, wait, oh, sorry.
You mean to go up?
Oh, no, no. Okay, I'm just gonna do, yeah, let me take another screenshot here. Okay, yeah, I'll take a look at this, after our session. But yeah, you're definitely not going to get anything like this difficult. In terms of like anything with a dihybrid cross, like that's going to be something that can be extremely time consuming. So either you get it, and in which case you eliminate what you can eliminate, and then you're left with 50-50,
Yeah.
or um it won't be given to you in that type of way. Like it would be given to you like, you know, in the section bank, there's a question about like red beetles and black beetles. I don't know if you've seen that yet, but that would be more along the lines of something that you would see that they would ask on the MCAT that has something to do with genetics. And it's mostly like a probability thing there, um, instead of like a dihybrid cross. Um, but yeah, I'll take I screenshotted it. I'll take a closer look. I just have some students until 8, but after 8, I'm gonna take a closer look at that.
Thanks. And when you email me whatever later, we can discuss about the next meeting time because I know you have a student right now.
Yep.
So just call it.
Okay. Awesome. So yeah, don't fret too much about this. It's going to be a very small fraction of your test. And probably a good thing to kind of like make an educated guess, flag, move on, just because it's so time consuming.
Yeah.
Yeah. And oh yeah, and then you're taking your next blueprint test. And
Monday.
have you been doing like the cars practice where we talked about like you checking that main idea stuff?
Yeah. Um, Usually my main idea lines up. I don't know in terms of timing.
Yep.
Yes, yesterday I did a passage for like 25 minutes. Like that's just way too much time.
Yeah, yeah. But how were your results?
I mean, it's like a little better. It's like 70%, I guess.
Yep.
But like, yeah, I mean, I'm obviously doing better. I have more time.
Uh-huh.
But like, my main idea usually lines up. I think it's more of just being able to find the more wrong answer. Like I end up with two answers, and then I sometimes, choose the wrong one.
Would you say you spend more time reading the passage than answering the questions, or vice versa?
I spend more time reading, probably.
Good. Okay. Okay.
But
Yep.
yeah,
All right. Got it. So yeah. Yeah. So I'll take a look at this and then I'll send you an email, and then whatever questions you had, you were gonna email me about that, I guess, right?
yeah.
Okay. Do you want me to put you in for next time?
I mean, we can email about if you want. But I was gonna say Sunday.
Yep. Can you do, let's see, 6 p.m. Sunday Eastern?
Yeah.
The 7th?
Mm-hmm.
Okay. All right, so 6 p.m. Eastern on Sunday the 7th. I will see you then, okay?
Yeah.
All right, awesome. All right, take care, okay?
Yeah.
Be good 4th of July.
Me too. And email me how much I owe you, please.
Oh, yeah. All right, I will. All right.